Thanks everyone for being here. We're really excited to kick off the seminar series for the new year with Professor Matthias Steinrücken, who's a member of the NITMB faculty and is also an assistant professor of ecology, evolution, and human genetics at the university. A full professor.
In July, and associate professor. But thanks.
At the University of Chicago, we're really excited to hear.
Thanks. Yeah, thanks for the invitation. Good morning, everybody. Thanks for being here. Today, I wanted to talk a bit about some work I've been doing recently with one of my PhD students, Adam Fine, and one of my postdocs, Xiaohang Shang, and Maren will also be featured in the end, about inferring selection from temporal allele frequency data. And to kind of get started, what I mean by that, what is the problem, just to introduce it, I have put up here this figure from this review article by Fan et al. In 2016, which shows a map of the world. And basically what this figure is supposed to show us is that modern humans originated somewhere in Africa. The pointer does not work well. Modern humans originated somewhere in Africa, but then spread throughout the world. And in all these different places in the world, they encountered different environments. And so if you think about genetics, if some people in the population, some individuals have genetic variation that helps them better to cope better with the environment they encounter, then living there for a couple of generations, this genetic variation tends to increase in frequency because it helps the individuals to better cope with the environment and makes them more fit in the lingo of population genetics. And so this map shows some examples of where genetics has helped us a lot to study these type of effects where genetic variation seem to have been beneficial in a certain environment and increased in frequency in the population. And a lot of these things there's like a lot of, wherever there's mountains, there's high altitude adaptation. A lot of this is also dietary. Some of it is also in response to some pathogens. And genetics has helped a lot in uncovering a lot of these stories and finding out about the genetic mechanism underlying these responses to these environments. But a lot of this work has been done often using basically contemporary genomic data from individuals that live in these places, and then looking for signatures that these past adaptive events left behind. There's like a lot of good theory and a lot of good methods that tells you what signatures you have to look for, and they work pretty well and have helped a lot. But in recent years, people have been starting to collect, in humans especially, but also other species, what they call this ancient DNA. It's basically, people have figured out how to dig up bones from people that died a couple of thousand years ago and extract genetic information from these bones. And so here I just have this nice picture from this article. There's a lot of articles, but I like this picture by Damgard et al., Ancient Human Genomes Across the Eurasian Steppe. So basically that shows us they were able to find some of these ancient remains in these different places at these different times and were able to extract genetic material. And for this process that I'm interested in of this adaptation, this selection, changing allele frequencies in the population over time in response to fitness differences, actually being able to observe the genetic composition of the population over time is incredibly helpful when you study this. Comes with its own challenges, but it is a different perspective than just trying to guess from present genetic variation what happened in the past. So this is basically what I'm interested in and what I'm going to talk about today. Using this type of data, observing, sampling allele frequency changes over time and figuring out what does this tell us about the underlying mechanisms, the underlying processes, population genetic processes that are going on. To be a bit more specific, to frame the question a bit more manageable, focus in a bit. When I talk about temporal allele frequency data, in a lot of cases, people are referring to genetic samples for biallelic locus at different points in time. So you just in the most focused, simplest case, you say, okay, we have this one genetic locus. There's two alleles, so there's two genetic variants at these locus in the population. And we have samples over time where we know that the individuals that we sampled have either one of these two genetic variants. And so I already, my introductory story had some examples, but there are other places where we can encounter this type of data. So some places are, for example, what people call experimental evolution or evolve and resequence. experiments where they basically have a laboratory population of here Drosophila or some other critters and put some stress on them, some new environment. And then they let them evolve or like let them live there for a couple of generations. And every now and then sequence some of the individuals to figure out what is the genetic composition of this population and how does it change after a couple of generations. And so this is some data from this people, Baghi et al., who did an experiment like this. They did a lot of sequencing and have a lot of data. This is just one specific genetic locus, one single nucleotide polymorphism, where there's two alleles. And I show you here, I plotted the frequency of this focal allele that, as you see, decreases over time. They have a lot of this data, but I just wanted to show an example here. And I'm going to get back to this. This particular SNP is not, I didn't choose this randomly, but I'll get back to this.
Or what, like, meaning why have, I'm not asking like what specific gene, I'm asking just why are you calling it that, like what is a focal allele?
It's basically, in this case, it's a single nucleotide polymorphism. There's this one position in the genome, this SNP6, where there's two possible nucleotides segregating in the population. And so these are two nucleotides that I don't know which ones they are, but there's like one nucleotide and the other nucleotide. So you look at this one specific genetic position and you see two nucleotides, and this is how they change in frequency over time. But the genomes are big, so they have a lot of data. They look like at a lot of specific single nucleotide polymorphisms.
There are two, and one goes away.
Yeah, exactly.
So I've seen many studies like this where you have some external stress, right, and then the genetics and things like this. Are people doing things where you have multiple stressors that are interacting?
Yeah.
4 or 5 stressors that you're trying.
Wait, are you asking whether people do that? I figure people do that. I'm not too versed in that literature, so I think people. I can see people doing both approaches. One, you want to isolate one stressor and you want to really know what does this one do, so you would only change one thing. But of course, it would also be interesting to potentially look at like a combination of things that happen and then try to disentangle what happens there. So I think I could see both approaches being done. We get to that. Just for now, this is just an example of data at this point, but we'll get to that. Not in depth, but I'll get back to this in a bit. Other data this is kind of ancient DNA already, where people extracted genetic material from diseased individuals. In this particular case, this is a data set that has been around for a while, and a lot of people have actually used this to showcase their methods. It's at this ASIP locus in horses, which there's kind of two genetic variants at this locus. In this case, it's actually kind of a gene on two different variants of that gene. And this locus is involved in coat coloration, I think in horses, but in a lot of other animals as well. And so again, they were able to extract this information that there's like these two alleles. And initially there's only one, but then over time, a second one shows up and does this dynamics. And as I already alluded to in the beginning, humans, there's a lot of this ancient DNA around nowadays too. Here at a specific locus is this kind of famous LCT MCM6 locus. This is a genetic variant that is supposed to, that allows adults to digest lactose. So usually you can't digest lactose if you're an adult, but this genetic variant allows you to digest lactose. And the story usually goes that once pastoralism became a thing, it was beneficial to be able to drink milk when you're an adult. And so in Europeans, there are some genetic variants that show this pattern where initially everybody could not drink milk as an adult, but then these genetic variants show up that allow you to do that, and they are very beneficial, and thus they go up in frequency. And I come back to this as well. And specifically, I'm going to be a bit more specific about what exact data that is. And so there are some other examples where you can encounter this type of temporal genetic data. Bacterial lab strains, there's this experiment, maybe some of you might have heard of this. Richard Lenski started that, I think, 30, 40 years ago, where he just started to grow bacteria in the lab. And did this for like, I think there are 90,000 generations or something by now. And there was some recent very nice work where they actually were able to then sequence some of these very old replicate and over time and interpret these allele frequency changes. And something else would also be viral populations. If you have like a patient that's infected by a virus, then you take like samples of that virus and assess the genetic composition at certain stages of the diseases or at certain points in time, and you can then kind of infer something about how the virus maybe adapts to the host and what exactly happens. So this is just my slide showing that there's a lot of places where we could encounter these type of data sets and where we would be interested in understanding what's going on. And so what's going on, that's kind of the key question that I want to talk a bit about today, is there adaptation to selective pressure? So we have these samples allele frequencies over time. There's like there's a lot of stochasticity that goes into all of this. So there will be some variation in the data that we see. And so the question is, the variation that we see, can this be explained by neutral dynamic, or do we have to invoke selection? So is that what we see just random noise, or is there really some selective adaptive force going on that's like responding to an environment or something? And to address this question, so today, my objective is basically to talk about how we developed a model to infer the strengths of selection from given data and characterize this underlying process. And so to get there, this is just like a cartoon version of what the data is. The data is basically, we have time going to the right here. And then at several time points, we take samples from the population. We sample a certain number of individuals. And we see, okay, at this biallelic locus, in our sample of 4 at this time, 3 individuals have the focal allele, 1 individual has the other allele. Then sometime later, we sample 3 individuals. There's 1 that has the focal allele, 2 don't have the focal allele. And then we sample another one sometimes later. So this is kind of the data that we have that we want to work with. Samples over time. So if we want to do anything with this, we have to kind of come up with a stochastic model of how this data originated. We have to come up with a generative model for the data. And so the model people usually use for this data is to say, okay, these samples originated from some population. So we sampled from some population. In the whole population, this allele that we are looking at was at a certain frequency in the whole population. And then these samples are basically noisy observations of this population allele frequency. And so there's a lot of randomness in all of this that we have to account for. And so this is kind of the randomness that's there. Initially, at some time t0, the frequency of the allele in the population was at a certain value. Then it underwent some dynamic, some fluctuations until the time of the first sample. So then at the time of the first sample, it was at that frequency. Then we took the sample. So we made this noisy observation of the population allele frequency. And then the allele did some more dynamics up to T2, was at a different frequency at T2. Again, we take a noisy observation of that allele frequency, and so on, until we have our sample generated. So this is our generative model for the data. We had this underlying population allele frequency that underwent its fluctuations, and we have then our sample are these noisy observations of that. But we do have, or that's not a problem, but what we have to consider when we want to understand this is that this is a possible frequency of the allele, and we took our noisy samples. But since they are noisy observations, this could have also been the underlying frequency. This could have also been the underlying frequency. We don't know that. So in order to understand the process that's underlying is, what we want to do is, we want to do likelihood-based inference about the underlying process. And what we need to do for that is, we have to basically compute the likelihood of observing our data, which entails integrating over all possible underlying allele frequency trajectories. And that does sound like a tall order, but it's actually manageable. And the way people made this manageable is this framework of hidden Markov models that has been used a lot in computational biology in different places. So we want to compute the likelihood of the data. So we want to compute the likelihood of observing the data that we see under some model. And for that, we have to integrate over all possible allele frequency trajectories. And in this particular framework, Bauback et al. Were the first ones to introduce this type of Markov model framework to formalize this. And as I said, this hidden Markov model framework allows you to integrate efficiently over all these hidden allele frequency trajectories. And in these hidden Markov model frameworks, you need to basically say two things. You need to say what the hidden state is and what the observation is, the emission. And in this specific case, and I'm going to get a bit more into the detail, the hidden states are the population allele frequencies at the sampling times. And the emissions, the observations, are the data we observed at time tk. So basically, like this. So our hidden states are the allele frequency at a given time, and our observations are the samples. And so then, in order to integrate efficiently over all these possible allele frequencies, this hidden Markov model framework basically chops this up into steps. You say, okay, we have some distribution of the population allele frequency in the beginning. We might know what that is. It might be a fixed frequency, then it's kind of a Dirac delta at some point, or it might be some distribution. We might actually be able to infer this, or we can make some assumptions. So I consider this kind of given for now. So then what we have to do is we have to figure out how can we integrate over the likelihood of going from any frequency, any possible frequency here to any possible frequency here. Then we can sum this over all the possibilities that we had here to get a new distribution at T1. What are the likelihoods to be at certain frequencies at time T1? What is the probability distribution of the hidden allele frequencies at time T1? Then we take the sample, get back to that in a second. Then we have to do the same thing again. We have this distribution of possible underlying allele frequency trajectories. We have to figure out what is the likelihood that we go from one frequency to another frequency, sum this over all the possibilities, and we get here. And then again, we take the sample. And again, we have this summing over the allele frequency trajectories, take the sample, and then we actually have computed the likelihood. So this is now what we want to do. We want to compute this likelihood using this hidden Markov model framework and doing these steps like figuring out what is the initial distribution, what's the distribution of likelihoods to go from a certain frequency to another frequency, and how do we incorporate the samples. And so this is how this works in this hidden Markov model framework in this case. I actually start with the emissions, the observed data, because that is more easy. Because if the population allele frequency trajectory is at x, so if you condition on the population allele frequency at a given time, then the likelihood of drawing a sample and observing a certain number of focal alleles is just a binomial. It's just a coin flip. Each individual can or cannot have the focal allele, and the likelihood that each individual has the focal allele is just given by the underlying population allele frequency. So that's actually fairly easy in this case. The more involved thing here is the hidden states, the population allele frequencies. How do we figure out the dynamics, or how do we model the dynamics of the hidden population allele frequencies? That's a random process, so they fluctuate randomly. So how do we describe this random process? How do we manage computing something with this random process? And a key ingredient here is the so-called transition density, or transition density function, which I denote here by PTXY, which is basically telling you the likelihood of an allele frequency change from x to y in time t. So this basically tells you if your frequency is x at a certain time, how likely is it that after time t, it's at y? So it gives you this probability distribution. And crucially, this is where selection comes into play. Because if there's no selection going on, we would kind of expect the allele frequency not to change. Whereas if there is some selective pressure that tends to increase the allele frequency, then we have a different expectation of how the allele frequency should change. So this transition density is where selection comes in, and which allows us to kind of model selection in this model and understand something about selection. So this is where selection comes into the picture. And so I want to take a few seconds to talk about what is this transition density function? How do we model that? How do people usually model these type of things? And the crucial thing is that's where selection comes in. So the key or the steps to get to this transition density function is to first think about what would we expect how the allele frequency changes from one generation to the next, under selection or neutrality. But I'm going to talk about selection. Neutrality will be just falling out. So we have two alleles at this locus. Call them arbitrarily A and small a and capital A. We want to keep this a bit more general than is usually done in the literature. We want to keep this as a general diploid model of selection. What that means is basically that diploid individuals, like humans, at each locus, you have two copies of an allele. And we want to kind of say, these two copies all can have an independent fitness advantage or disadvantage. So we say, if our diploid individual has two copies of the small a allele, that's the baseline of 1. If the individual has one copy of the capital A allele, it gets a fitness benefit, or the opposite of a benefit, S1. And if it has the type has two copies of the capital A allele, it gets this S2. And without going too much into the detail, if you denote by X the population of the allele frequency, of the in the population of the capital A allele, then you get that this is the expected change. You should expect this. This should describes what you should expect under this fitness model, how your allele should change from one generation to the next. And without going too much into detail, there's this very characteristic x1 times 1 minus x that shows up in a lot of places, which is kind of the variance to some degree. So it gets stronger, selection gets stronger, the variance is higher. And then there's some dependence on these selection coefficients. And so this allows us, if I tell you what is the fitness model that I have, which can be parameterized by these two numbers, then this is how we should expect the allele frequency to change. In the literature, people use almost exclusively the so-called additive model, where S1 is 1/2 S2, where basically the fitness of this type is intermediate between these two types. And that is basically a simplified model, and it's called additive because basically the number of capital A alleles determines how fit you are. And the reason why this is used almost exclusively in the literature is that this maybe more complicated formula simplifies to this. And it makes a lot of the math easier. And so that's why people prefer or use that a lot, because it makes the math easier, not because it's maybe more appropriate for the situation they're interested in. But as I said, we want to kind of keep this a bit more general. And so we're going to deal with this. And the reason, some reasons why this is interesting is because this allows us to model dominance and recessive fitness effects. So you might remember from some biology class you once had that dominance means, in this case, just having one capital A allele gives you the full fitness advantage already. So then these two are equally fit and are fitter than this guy. So then S1 is equal to S2. And recessive means you have to have two capital A alleles to get the fitness advantage, and you get no fitness advantage in any other case. So then S1 is 0, and basically this is the only one that gets the fitness advantage. And so this kind of model allows us to be a bit more flexible and understand selection in some of these more, in these other situations that are not only this additive model. I want to mention this here because I come back to this in a later briefly. These are kind of, if you think about it as being a two-dimensional fitness space, S1, S2, these are basically one-dimensional, one-parameter subspaces of that. Yeah, so we want to keep this a bit more general at this point. But anyway, regardless of what exactly we do, once we have the expected allele frequency change, this is how the stochastic fluctuation around this expectation are modeled in population genetics. Getting back to this transition density, PTXY, what's the likelihood to go from X to Y in time t? People usually use this model called the Wright-Fisher diffusion to model this, where you say the change over one generation is approximately normal distributed, where the mean of that normal distribution is the expected allele frequency change, this mu, from here. And the variance, so the mean is the expected allele frequency change. Oh, I forgot to mention, neutrality is easily in here. If we put all these parameters to 0, that means everybody's equally fit. That's the neutral case. And we get expected change 0. So that's in there. So, yeah. Say again?
Your selection is linear in X, right?
Linear in X.
Yeah, so you have S2.
This is a polynomial of third degree. That's not linear in X. Okay, I think maybe what an answer to your question is that this is still kind of not all the selection you can ever imagine. You could, in principle, make this frequency dependent. You could say, I mean, the expected change in the mean does depend on the frequency of x, but you could make it more complicated. You could make a more complicated frequency dependency. You can. I'm not going to do this today because this is used a lot in the literature because it makes the math easy. This makes the math intermediate. An arbitrary fitness function makes it arbitrarily hard. So I'm going to use this model here. But yes, you can make this, you can model other situations of frequency-dependent selection in this framework, but then the function on the right-hand side here is what you have to choose, basically. You have to tell me, how does the frequency change? How would you expect the change to be, depending on the frequency that is currently at? But yeah, you can put an arbitrarily complicated function here. Just this has some of this motivation behind it, that it models these kind of known cases of additive selection or dominance or recessive. So this is kind of a manageable parameterized version of this model where selection does, there are a couple of cases of selection in there that you are interested in, but there are others you could be interested in that is not modeled by this, but you could put it in here. In principle, the Wright-Fisher diffusion framework wouldn't change, it would just the mean here would change. Yeah.
That you got from the way that you got from essentially your probabilities part to your expected.
Yeah.
Like basically writing down master equations of transition probabilities and then calculating an expectation.
Yeah, I wouldn't say you need to invoke the master equation. It's kind of more a bit. Actually, Luis just gave a lecture about this a couple of days ago. You basically figure out, like, if you have the yeah, you say you just play out this stochastic experiment. I have these types in the population at certain frequencies. I have these probabilities to choose them. Then what would I expect how the frequency changes from one generation to the next? So it's just like working through the math of this kind of experiment. Okay, so we have some expected allele frequency change. That's the mean in this normal distribution. And in population genetics, how people model this, or in this Wright-Fisher diffusion framework, how people model the stochastic variability around this mean, like how much does this deviate from this mean? Model this by this variance in this normal distribution, which almost always has this characteristic form of x times 1 minus x divided by the effective population size. And this is called genetic drift because it shows up in a lot of places. It got its own name. So this Wright-Fisher diffusion then allows us to say what this is. We say, okay, each one generation step is approximately normal distributed with this mean and this variance. And so if our t is like several generations, we just chain a bunch of these normal steps together. And so this then allows us to figure out what this PTXY is. And maybe I can say this already here. There are kind of different ways to write this down. A very common way is to use like a Kolmogorov forward or backward equation to write down like a PDE for this transition density function, and then possibly solve that equation. Yeah, so we have, without me having presented all the detail, we can say what this is, how the stochasticity of this evolves. Yeah.
Steps and also going back. I think there's a limit, right? You're taking a limit from a process that was explicitly discrete, and you had. Okay. I brushed a little bit about that. If you exactly, so kind of the limit is, in a sense, if you have a finite population with a finite number of individuals, you can write down to a degree how the transitions would be very complicated, but you can take the limit of an infinite population size to get to this Wright-Fisher diffusion. So that's kind of the limit. So it's the limit in an infinitely large population, and then the limit is used as an approximation for finite populations. Yes, so there is this limiting process. okay, so now we have PTX, and so we can basically implement this. We now know, given some initial frequency distribution, we know what are the likelihoods to go from here to here in a certain number of generations. We know the binomial likelihood of sampling these. We know then how do we do these other transitions. So we have now all the pieces together in place without me having given you all the details to compute likelihoods under this model, to integrate stepwise over all these possible allele frequency trajectories and compute the likelihood of the data. So there's different implementations in the literature where people have done this. So the question is now, okay, I've told you basically what the model is, how the model works, what the dynamic of the model is, but that's not immediately telling you how to implement that likelihood computation efficiently. So that's kind of where some progress has been made or where people are working on to some degree. So one thing that Bobak et al. Already did when they introduced this hidden Markov model framework in this situation, they used only this additive selection model, but what they did is basically looking at this Kolmogorov forward equation, discretizing this allele frequency space 0 and 1, the possible allele frequencies, and solved the PDE transition, the Kolmogorov forward equation using a finite difference screen numerically, and use that then in this whole hidden Markov model framework to compute the likelihood of all the transitions, included the observation of the data, and use this to compute the likelihood in the model. I have done some things in this space to compute this efficiently. Actually, this one is already a while ago, where we considered this more general diploid selection model. And we used a different numerical procedure to compute the transition density function. And without going too much into the details, although I figure if I ever could go into the detail, this might be an audience where I could do that. What we did to compute this transition density function is we used the eigenfunctions obtained from the spectral decomposition of the diffusion generator, or the Kolmogorov backward equation, which is basically similar to solving a PDE using Fourier basis, just like with a slightly more complicated basis. So we devised this scheme to compute numerically this transition density function. It actually turns out if you put this into some of these hidden Markov model equations, it actually simplifies a few things nicely, so that in the end you end up with some pretty nice dynamic program to compute the likelihood. So we worked out kind of how to compute the likelihood using this approach, which works well for long times and moderate selection. But to be fair, from an applied perspective, it's computationally pretty demanding for short times and strong selection. Because basically, if you have this normal distribution for the transition density, if you go very short times, that becomes very peaked, almost like a Dirac delta. And then this corresponds to expressing a Dirac delta in a Fourier basis, where you need many, many terms to get it hopefully reasonably accurate. So that is basically the problem, what makes this challenging for strong selection and short times, that you have this numerical thing that you need a lot of bases, and then it gets computationally very demanding. But for longer times, you need less bases, and then things work nice enough.
So the problem that it's not a good approximation, or you just need like a lot of.
Yeah, it's the latter, that you just need. I mean, the approximation is in the limit of infinite terms exact, and you need more and more terms to get it get the accuracy that you need in applications. So it's actually you need like invoke like some high precision numerical libraries, which makes it like your standard doubles don't cut it anymore, so that just makes it harder. But I do like this approach a lot, because it's like the theory behind this I think is very, very nice and very sleek. But yeah, from an applied perspective, it's unfortunately a bit challenging, or not always applicable. So that's why I did some more recent work with some of my students and postdocs on this, specifically in this paper with Xiaohang. We just said, okay, let's maybe just do the very naive thing. Like maybe the very naive thing is actually pretty good. So what we did is just we again looked at general diploid selection. We discretized the allele frequency space. And then we just use these probabilities, the normal probabilities for the one generation step. So basically just saying, what's the probability to go from a certain frequency bin to a certain other frequency bin? That's just given by the normal distribution. So just compute that probability under the normal distribution. And then you get these transition probabilities from certain frequencies to certain frequencies. And then you have it for one step. So if you have more generation steps, you just chain these probabilities one after the other. It's just like matrix multiplication. And we implemented some other tricks, computational tricks, to efficiently reuse some of the computation. And it actually turns out that this naive approach is actually fairly quick in simple situations. So we developed this method into like a publicly available software tool that allows you to compute likelihoods. If you give me S1 and S2, I can compute the likelihood of the data for you. And then you can compute this on a grid and find the maximum likelihood estimator for your selection coefficient. Yeah, I guess I already alluded to this, but the problem is a bit the curse of dimensionality. If you have like only one parameter, like only additive, only one parameter, this works actually really fast and gives you really good results. But the more and more parameters you add, the more challenging it gets. Like already, the two-dimensional grid is already, you already need a bunch of time to get anywhere there. But it works well in simple situations and is pretty fast. Yeah.
I think I'm not understanding the full complexity of it, but it seemed to me that that's exactly what you're doing, is what should be done. And once you're Gaussian, then you can just iterate the Gaussian to get another Gaussian.
Yeah, no, wait. But the problem is that the Wright-Fisher diffusion is only infinitesimally Gaussian.
Yeah.
It's not, because of the variance is. It's really not. It's like not good to use a Gaussian for a longer time.
Yeah, it's a path integral of the exponent.
Yes, I mean, yes, it is a path integral. All of these are just numerical ways to compute the path integral, and the goal is to make this like quick enough to be done in applications on a large scale, because you have huge genomes, so you want to do it on a large scale. But yeah, it is an approach to compute the likelihood approximately. So as are others. Yeah. So, but yeah, I guess the setup is a bit that people were doing very elaborate stuff to do this. And then we were just like, well, what if we just, like, nobody ever did the naive thing. And so it works actually pretty well. But as I said, if you have like more parameters, it gets harder, because like grid-based optimization in larger, higher dimensional spaces there's like some diminishing return and stuff. And okay, I get to this maybe in a second. Actually, maybe here already. So we worked on some other approach with Adam Fine, one of my PhD students, where we basically modeled this very similar. We said, okay, let's discretize the Lie frequency space, do the Gaussian transition density for the steps. But for the likelihood optimization, we didn't do grid-based optimization. We implemented some expectation maximization approach to find the maximum likelihood estimates of the parameters. And the thing is, in this, if you set this up correctly, there's some nice theory you can do to solve the M step analytically, which is some extension of people that did it before in the additive case. And the advantage of this is over the grid-based optimization is that this works in higher dimensional scenarios better than this grid-based stuff. And you might ask correctly, I only have 2 parameters in this model, but I could have a more elaborate frequency-dependent selection model with more parameters. One could maybe do something there too. Other parameters that we were actually interested in doing as well is estimating the initial frequency, or like a parameterized distribution of the initial frequency. So we actually use this to estimate the initial frequency, which using this approach would have become cumbersome and prohibitive. Okay, so this is basically how we can compute likelihoods and do optimization. It's the maximization step. So it's just, I don't know. No, I don't want to go too much into detail, but this expectation maximization is basically this iterative procedure where you start with a guess for your parameters, you compute an expectation, you realize your parameters are not the best, so you maximize them. Then you have new parameters, you do another expectation. So it's like this iteration of EM steps. And the M step has like some nice properties in this specific problem and allows you to do that a bit more efficiently. Yeah, so now we can do some optimization and compute likelihoods. So why not do that? So here, I'm just going to go through the data sets I showed you in the beginning and show you, okay, we can use our methods to compute a likelihood surface in S1 and S2. So we have this data. We compute likelihoods for different values of S1, S2 under some, we fix some of the values to some numbers. And we get that the maximum of this surface is here where the red point is. And not surprisingly, that tells us S1 is negative, S2 is negative. So not surprisingly, you saw this go down. So makes sense that we get some S1 and S2 negative in this case. So I mentioned in the beginning, I didn't choose this data set randomly. This was one SNP from a gene that showed up in the original study as one of the strongest hits. Just wanted to have something to show, like there's some selection going on potentially. And maybe there's some other people that know better about Drosophila genetics, why it makes sense that iron-sulfur cluster binding shows up as a response to heat stress. Yeah, but we can use the data and compute likelihood surfaces like this. This is for the ASIP locus, where this is the data, this is the likelihood surface. We get, interestingly, without going too much into detail, but this suggests that S2 is like pretty small or maybe even zero-ish, and S1 is some positive number. And if you think back to how I defined the likelihood model, that is a model where the mixed genotype is the fittest over the ones that don't have both alleles. And that is kind of a model of balancing selection, some force that keeps both alleles around. It is fit to have, it is good to have both alleles in the population. It's not good to have either one only around. And it makes some sense that a model like this shows up here because you see, initially we only have one allele, then a second shows up, but at the end, both of them are kind of around. So it makes sense that we get something from the model that tells us, that favors a model where both alleles are around. Yeah.
Why is the effective population of horses much larger than that of flies?
Because the flies are in the lab and the horses lived in the wild.
Does this value of S1 and S2 predict why the photoallele peaks a little bit before it starts to rebalance?
No. I think what the model is telling you that this peak and rebound is noise in the sampling. Like if you think about for given values of S1 and S2, the model would predict a smooth curve. But what the model is basically fitting is, I expect this smooth curve with some noisiness around it due to the sample. But then on top of that, I have this random binomial sampling, and that actually can account for a bit of noise. Like that, even if your frequency is 40%, you might see 60 in your sample if you only sample 10 individuals. That can happen. That's not impossible. So the model is kind of trying to account for all of that and give you its best guess. But yeah, if you would use just the S1 and S2 to predict a frequency trajectory, you would get a smooth curve. If it's like not time changing or not. Like there are other models of selection where you could mess around with that. But in this case, you would get a smooth curve, and some rebound and peaking is likely just explained by the model to be noise, basically. Okay, and then lastly, I wanted to also show you quickly, and I get back to this actually, the LCT locus in human that I showed you in the beginning, this lactate persistence allele, where again, we have none of the focal alleles in the beginning, but then it shows up and suddenly everybody has it, or nearly everybody has it. And so obviously S1 and S2 are like both positive because there's like some positive selection for this. But interestingly, so I told you in the beginning, these dominance, recessive, and additive are like one-dimensional subspaces. And you might have noticed that I put some lines here. So this is the additive subspace. This is the dominant subspace. And this is one of the other ones. Okay, let's keep dominant and additive for now. So these are the two one-dimensional subspaces. And this maximum does go a bit towards the dominance line. So this might suggest that there's actually dominance. And there's some evidence in the literature that the genetic mechanism of this genetic variant is actually dominant. And so this maybe makes some sense in the context of what we know about this specific gene. So I've shown you that we can take these data sets and compute some likelihood, and then we can talk a bit about the likelihood surface. But that's not really genuine if we don't do some statistics to validate that our method actually does what we think it does. So we did some quite extensive simulation study to assess the accuracy and characterize how well our method works in certain situations. So we simulated some populations with some population size, sampled a bunch of individuals, a bunch of generations for some time, started at a given initial frequency, simulated different modes of selection, additive dominance, these two modes where the middle, the heterozygote, the mixed genotype is either the fittest or the least fit, and for several selection coefficients. So we simulated a bunch of data. We simulated a bunch of other scenarios as well. This is just what I'm going to show you. And in the analysis, we estimated the selection coefficients and the initial frequency. However, if you don't want to estimate the initial frequency, we realized that assuming a uniform distribution actually works fairly well. So if you don't know it or don't want to estimate it, it's actually kind of okay to use the uniform distribution. And this is something that I also wanted to mention is we actually found that optimization in the full two-dimensional space is actually a bit unstable. And the reason for that is, I mean, the likelihood surfaces are sometimes flat in some direction that makes it really hard to navigate this two-dimensional space. So we actually found it way more stable to estimate parameters only in this bespoke one-parameter subspaces, and then compare likelihoods of those to figure out what is actually the right mode that we have to use. Okay, so then the first question we would ask from this simulation is to say, if you simulate neutral replicates, compute likelihoods, do some standard likelihood ratio testing, like the likelihood of neutrality versus the likelihood under selection. Is the likelihood increase enough to call it selection, to reject the null hypothesis? So if we do that and compute p-values based on this, the fact that everything lies on this x equal y line tells us they are well calibrated. We only reject neutrality, or we don't reject neutrality if it is actually neutrality. We don't do it in too many cases. So this tells us everything actually looks pretty good if we think about rejecting neutrality as a null hypothesis. So then the next question is, if we do simulate under selection, can we figure out that it is selection? Do we reject the null hypothesis if we simulate it under selection? So we simulate it under these different modes of selection with different strengths, and we ask, can we distinguish selection from neutrality? And a common thing people do in statistics is compute this thing area under the curve, which basically tells you if this is 1/2, you're guessing randomly. And if this is 1, you're perfectly in classifying the two things. And not unexpectedly, if selection is somewhat weak, we are not very good at distinguishing because the data looks like neutral because there isn't a lot of selection going on. But as selection gets higher, we're actually getting pretty good at classifying and identifying selection if there is selection. So this works pretty well if selection is strong enough, where strong in quotation depends on your scenario, obviously. If you don't have a lot of samples, you can't say a lot. Or if your samples are very far apart, you can't say a lot about something that happens in a short time. Or if you only sample very close together, you can't say stuff about something that happens over long times. So what strong means depends on what specific scenario you're interested in. But as I said, we simulated a bunch of scenarios, and basically our recommendation would be, if you have some data of this type, simulate data that fits that scenario to figure out what you can do with your data. Okay, so if selection is strong enough, we can identify it pretty well. And then we can actually look at the point estimates. So if we simulate under certain true selection coefficients, how well can we estimate them? And we do pretty well. The mean is pretty well in the aligns pretty well with not a lot of variability, although the over and under dominance, they show a bit more variability in this case. But yeah, as I said, this framework allows us to characterize, like if you have a given scenario, simulate data in that scenario, And this all tells you what can you do in your scenario and how well can you trust your estimates. And so then lastly, with the simulated data, we also can play this game to actually figure out what is the mode of selection, where we simulate data under neutral, additive, dominant, recessive, and we forget about what we simulated it under, and we ask the method, please tell me, what do you think this was simulated under? And so if it would be perfect, there would be ones on the diagonal here. And there's like some 0.8, 0.7, 0.6 on the diagonal. So we're doing okay. It's not perfect. Again, the stronger selection is, the better this is. So if selection is not as strong, we don't do fairly well. So actually, our kind of conclusion in this respect is that you can do this and look at it, but you should treat it like with a bit of caution. It's not fully reliable in any case. Okay, so now that we have done some simulations, we are kind of confident that we know what our method can do, and we know when we can trust it and when we shouldn't trust it. So let's apply this to some real data. So we wanted to apply this to some real data from humans. But we have a slight problem there, and I'll get back to this in the Outlook. Our method assumes that the data comes from a single well-mixed population. Humans live on different continents, and these samples come from different continents. So the assumption of a single well-mixed population is likely wrong for any human data set. But maybe we can do something and say, but maybe it holds approximately. So we follow some rationale from some previous paper where they said, okay, where could we expect this to maybe hold, that it's a single isolated well-mixed population? So what they say is, okay, Great Britain is an island. That's pretty isolated. That's pretty good. And there's also some work that shows that the last major population turnover was some 4,000 years before, 4,500 years before present. So if we only consider samples past that are more recent than this from Great Britain, maybe our assumption of a single well-mixing population is actually not too wrong. And so that's exactly what we did. We used this data set where people collected a lot of the ancient DNA that's publicly available and extracted individuals that were more recent than this population turnover. More specifically, this is kind of more of the specifics. We used this database filtered for the political entity UK, which gave us the samples on Great Britain. We filtered the date. We removed islands manually to just have the mainland. And so we're left with 500 individuals that are sampled there at that time. And we did some quality controlling so that we don't analyze data that we shouldn't analyze. And then, so this is effectively samples. They are in this public database, but they were produced by these people. We also implemented a heuristic to account for variable population size. And we analyzed the data under all modes of selection, but here I primarily show you additive just to not keep you here forever. So this is what we get. We get this Manhattan plot where we basically plot the log p-value. And just to remind you, in this plot, a large number is evidence for selection. So what we see is, we see this here. There's like one cluster of strong p-values. And we actually should have seen that, because if we didn't see that, our method wouldn't work. So this is actually the lactase gene. This is this genetic variant that confers fitness advantage, or where you can digest lactose into adulthood. And this is kind of one of the strongest signals of selection in Europeans in recent years. So if you don't find that, then you did something wrong. So we're happy to find that. But then the question is like, what about the other stuff? There's like a lot of other stuff that's kind of above the genome-wide significance line, but we weren't sure whether we can trust all of that, whether we really should say every point that's above the line is a significant signal. And because I'm running a bit out of time, I'm not going to go too much into detail, but we thought a bit about looking at this in more detail and realized that sometimes it's a situation where there's significant hits and the surrounding area shows some support. And other times there's a significant hit, but the surrounding ones show no support. And under population genetics processes, you should expect that picture for a real signal and this for some artifact in the data or some less trustworthy signal. So we developed some approach to kind of correct for this and figure out when do we have surrounding support, when do we not have surrounding support, and basically then ended up with the rather that, okay, we trust LCT, and we trust these 5 others, and the others seem to be some less supported signals. So we didn't trust it to be conservative. Maybe there's some room for following up. And these 4 have been identified in the literature before as signals of selection in similar or other data sets using other approaches. And this one is actually one that we found in this study, which is, I think it's a genetic variant that regulates iron in the blood and is associated with some disease phenotype. So maybe there's like a story to be told about that type of stuff being relevant in these populations in Great Britain. Okay, so with this, I just wanted to kind of wrap up and say, okay, what are some of the next steps? As I said, when I introduced this data set from Great Britain, it is really not a good assumption to assume a panmictic population for humans. There will be some population structure. So there are a bunch of approaches I'm thinking about trying to deal with that. Other things to think about is selection varying over time. So it's not this constant process, but yeah, more complicated models of selection, maybe more realistic ones. And lastly, I also think there's some nice applications to understand direction and stabilizing selection. There's some more challenges that come with this. One that Maren worked in her PhD study on is that there's some population genetic effects that make this, like there are a lot of population genetic effects that make this complicated, and statistical effects, and Maren worked on some of them. But I do think there's some nice stuff to be done there. And so I'm just gonna wrap it up here. Thanks to everybody who has contributed, and thank you guys for being here.